Hey everybody, it's Professor Schott. Welcome back to another lecture. This time I'm going to try to go through all of Module 2 in one video. That way we don't have to break it up. And my throat is dry from talking so much, so please excuse me while I have sips of water. All right, so we are starting here. We're going to talk about Module 2. This is how do viruses evolve? Lesson 1, what is evolution? So the first questions that we're going to look at in this lesson are, what does the term evolution mean? And how does evolution relate to biology? And then last, we'll look at, are viruses alive? So again, we're relating the phenomenon that we saw with COVID-19, how we saw it jumping from an animal species into people, to evolution as a whole, because this is something we witnessed. So these are some skulls of different species of Homo that we are related to. So this is Homo neanderthalis, Homo antecessor. This is us, Homo sapiens, and then this is Homo erectus. So when people are presented with this idea here, it's not something that they witnessed, right? Like, we don't see Homo neanderthalis. We see the fossilized remains, or evidence that they existed, but it's very hard for the average person to understand this. So this is why looking at viruses gives us a little more context. So what is evolution? It's a unifying theme in biology. It refers to changes in a species or population over time. And so what we're looking at is simply that we have seen change over time in the history of our planet. So by studying and classifying life on our planet, scientists have been able to recognize that the organization of life provides evidence for change over time. So the question then isn't really, is evolution happening? We know it's happening, but what are the mechanisms of evolutionary change? So here, if we look at this tree over here, we can see that there are bacteria, or the domain bacteria, domain archaea, and then the domain of eukaryotes or eukarya, and how we all have a single common ancestor down here. So it shows that there's some kind of change that's happened over time and that we are all somewhat related. So by studying life on the planet, scientists have been able to recognize that the organization of life provides evidence of change over time. Evolution occurs at the population level. So we say that it doesn't occur at the individual level, it occurs at a population level. So the evolution of species only occurs when heritable changes in the genetic information, DNA or RNA of an organism, are passed on from generation to generation. So what this means is, if I were able to develop a trait in me that enhanced my survival, if I wasn't able to pass it on to my offspring, it would be moot. It doesn't matter. And it wouldn't be evolution. So evolution is when traits are passed from generation to generation, and they have a tendency to be better suited to the environment, if that makes any sense. So this will often allow the organisms to survive and reproduce better in the setting that they're in. So how does evolution relate to biology? When we talk about the history of life, we use billions of years ago, or MYA, millions of years ago. It's useful to think of life on Earth as a 24-hour clock. So the Earth is roughly 4.6 billion years old. And life on Earth here, so if you see 4.6 billion years ago, we see the oldest bacterial life here at about 4 billion years ago. The first photosynthetic organisms are at about 6 o'clock here, and that's our little marker. And then if we go all the way through here, we don't even have enough oxygen on the Earth to support life as we know it until about here, 1 p.m. The oldest eukaryotic fossils, so these are organisms that are similar to us, are here. on this side at about 5 p.m., 2 billion years ago. So, really, about 2 billion years go by where most of the life on Earth is prokaryotic, bacterial. Then we see the oldest multicellular fossils at 8 p.m. here at about 1 billion years ago. We see land plants emerge in here. Then we have the age of the dinosaurs here at about I'll have to check, but it's showing at 11 p.m. I think is what they're showing. From 10 to 11 p.m. was the age of the dinosaurs. And we don't even have us. First appearance of Homo sapiens. There we are, 11:59, or like 1 minute to midnight. So this, if you guys have ever been to the Houston Museum of Natural Science, they do have a fantastic paleontology hall, and you walk through the timeline there. So if you have been to the paleontology hall, they start at the formation of Earth, and then you walk through the exhibit, and you end at the appearance of Homo sapiens. But you get to walk through a lot of the age of the dinosaurs, which is really fun. But there are a lot of things before that too that you'll see in terms of fossils. So anyway, so this is proof that we've had change over time, and a lot of it is that we are looking at fossils, right? We know that at certain points in Earth's history, it did not have the life forms that we have today, but life existed. But what changed? How did that occur? So evolution is recognized as one of the unifying theories of biology. So the last thing we have here is, are viruses alive? Biologists use 5 characteristics to determine if something is considered alive or not. These include the ability to process energy and materials, maintain an internal environment, respond to stimuli from the environment, reproduce, and adapt to changing environmental conditions. So, when we look at viruses, they are not able to do any of these things unless they are in a host. Since they rely on a host for these, we have fairly well determined that viruses are non-living. But this idea has been challenged by a discovery of a new group of viruses called the giant viruses. Some of the giant viruses possess the ability to manufacture their own proteins, which is a characteristic that was traditionally reserved for organisms that are alive. All right, so we are closing out that one. Let me double check, or that section. Okay, so next I want to talk with you about lesson 2. So this one is, how does evolution happen? And we're going to be looking specifically at natural selection. Sorry, I'm double checking our study guide to make sure that we covered everything. One of the things we ask in the previous module is, what is evolution and its link to viruses, and how are viruses able to evolve rapidly? One of the things that viruses can do is they produce billions of offspring, or I guess you could say millions, um, really quickly. So they reproduce really, really fast, and they produce a lot of offspring in each kind of reproductive round. So what they do is they're going to infect your cell. They're going to make, thousands of copies of themselves. They're going to burst out of the cell. But when they do this, they make copies of their genome. Most viruses do not possess the genome checking, I guess you could say, hardware to see if they made errors or mistakes when they copied their genome. So that means they're taking their genome, whatever genes they have, and they're making a copy of it. But if they make an error, they don't bother fixing it. They just stick it in the baby virus and on it goes. So that means that when a virus reproduces, the error rate is really, really high. And sometimes this error rate can be advantageous. So sometimes there's an error that happens when it's copying its genome that allows it to infect a host. So if there's a whole bunch of them with that error and it can infect a new host, then it's got a new place to kind of go, right? But a lot of the errors can just be nothing. So maybe nothing happens. Or maybe it makes the virus not as good at doing what it did before. So there's all these changes. So the answer is viruses are able to evolve so rapidly because they have such a short generation time. When you think about us, the human population can't evolve quickly because we have a very long generation time. If you think about it, we don't produce that many offspring either. So evolution is going to happen from generation to generation to generation. And the pressures that are going to kind of determine what new things or traits that are evolved make it or are advantageous are going to be coming from pressures. So when we think about viruses, the pressures are our immune system. So they want to evade our immune system. And that's part of the reason why we see a lot of shift. If you notice, we have flu vaccines every year because influenza viruses mutate rapidly and they are novel or new to our immune systems. So this is how they evade being caught and ensure their own survival. So I wanted to touch on that one. Let's see. Okay, I think everything else is pretty explanatory in the module, but if you have questions, let me know. You're always welcome to do that or go back to the reading. Okay, so now we're going to go to lesson 2. How does evolution happen? And we're going to be looking at the mechanism of evolution. This is our main mechanism. It's called natural selection. This is the process through which populations of living organisms adapt and change. It was provided independently by both Charles Darwin and Alfred Russel Wallace. So they're kind of credited with this. A lot of people credit Darwin, but they were both doing it at the same time. Their explanation of evolutionary processes forever changed the science of biology. We have a couple of other contributors here. George Cuvier, he was a zoologist. He studied the fossil record and noticed that it showed a succession of different forms of life over time. He believed that all species were created at the exact same time and that catastrophic events had caused some to go extinct. So that was his explanation for the change in the fossil record that he saw. Erasmus Darwin is a naturalist and philosopher who proposed that evidence from animal development, animal breeding suggested that species may evolve over time. Then there was John Baptiste Lamarck. He was a naturalist who believed that the complex life forms descended from simpler forms. So he thought that over time, organisms would just get more and more and more complex. He proposed that individuals can acquire characteristics that enable them to evolve over time. And this idea is referred to as the inheritance of acquired characteristics. Then there's Charles Lyell. He was a geologist who proposed that Earth's surface is dynamic and constantly undergoing erosion and uplift. And this explained to him how the different fossil layers were formed. And then we have Thomas Malthus, who is an economist who stated that populations, in this case humans, sometimes increase at rates faster than their food supply can increase, and thus this creates competition or a pressure. So each of these guys contributed an idea. None of them were 100% right, but a lot of them contributed something to natural selection and the understanding of evolution. So Darwin, you may have heard of Darwin's finches. He rode around on a ship, the Beagle, and he went to the Galapagos Islands. And these Galapagos Islands, he noticed some really interesting things about the species living there. So he noticed that there were different finches here that had different beak shapes. So this is the large ground finch, and notice its wide, kind of fat beak. And then this is a woodpecker finch. It's a little more pointy. And then this is the common cactus finch. It looks a little more kind of like an intermediate between these two, right? So these observations of these finches and then also of the tortoises on the Galapagos provided him with ideas on how natural selection drives evolutionary change. And let me go back to this. What we find out through this is that these finches each have different food sources. So as you can imagine, a finch with a beak like this might eat bigger seeds. This one might eat smaller things, and this one might eat something kind of somewhere in the middle. And so he hypothesized that these guys may have once diverged at some point from a common ancestor, but that because of what they had as their food source, that there was some kind of pressure on them, and it drove this distinction between the three. So how does natural selection explain descent with modification? So we call this descent with modification, meaning that organisms descend, but then they have modification. So they're inheriting something, there's some kind of change. So individuals vary in their traits. Variation exists in a population for heritable traits. This does not just include physical traits such as beak shape, like what he saw, but also physiology and behavior. So what we have is when we have offspring, we're going to have offspring that have different traits. And those different traits are going to potentially give that offspring a advantage or a disadvantage, right? So individuals are struggling to exist. Populations often produce more offspring than can survive, and this creates competition for available resources. So in the case of the birds, we start out with maybe a population of finches that have some variability in their beaks, and they're all trying to compete for food sources, right? Individuals differ in their fitness. Individuals who have favorable traits will survive at greater rates than those without them. When these individuals reproduce, they pass these traits on to the next generation, and thus the term fitness can be used to describe this reproductive success. So when we talk about organisms being able to survive and reproduce, that is the end goal of everything in biology, and especially with natural selection. So an organism is considered fit if they survive to reproductive age and are able to produce or reproduce offspring, and especially that those offspring survive. So that's kind of the other key thing, because if you produce offspring, but they aren't very fit, then it doesn't matter. All of your genes end there. They're no longer passed on to your offspring or any further in the generations. Populations become adapted to the environment. So over time, natural selection increases the frequency of the advantageous trait in the population and decreases the frequency of detrimental traits. Over multiple generations, the population becomes adapted to their new environment. So to give an example of this, let me get my little marker. So let's pretend, let me use this. And this is a true story, but there was a population of mice, and we can say, here's one. And then we're going to do one in a different color. Let's say brown. We're going to pretend that this brown one is black. Okay. And these mice lived in an area where there is volcanic rock. Okay. So in the volcanic rock area, the organisms, the mice with darker color, are going to be less visible by predators. So let's say there's a hawk predator flying over and he's looking for his next meal. The darker ones will blend in better. Okay, so our black ones will blend in better. Our white ones will be much more visible. So let's say that this white one and this black one have babies. So then here's all their babies. And in their offspring, let's say that these are the worst mice ever, that there are 6 total, right? Here. So what's going to happen is if these mice stay in this area on the lava rock where it's dark, these are going to have a tendency, the white ones, to get eaten by the hawk. So over time, we'll end up with fewer and fewer white mice, and we might have brown mice, but the brown mice still carry a gene to make white mice babies. So in every generation, even if there's a brown mouse and a brown mouse and they have babies, they might have a couple of white mouse babies. But the same thing's going to keep happening. So we're going to maintain this gene in the population, but it's just going to be less frequency of it. We won't see it as much. Okay. So that's what we mean by individuals vary in their traits. So even something like this, where we might see more brown or dark mice than we do white mice, there's still going to be some variability in their traits. The individuals are going to produce more offspring than can survive, which is because they just do. That's kind of what mice do. They produce lots and lots of offspring at a time, and typically way more than can actually survive. And then they're going to compete for resources. So while these guys are out there competing and looking for food, this one's probably going to get seen, which means it might not make it to reproductive age, and it might get taken out. If it gets taken out and it doesn't make it to reproductive age, then it's not very fit for that area. But let's say we reverse this and there's a sandy area nearby that is lighter in color. So over here in the light side, the brown mice are more visible. I can't draw my white mouse because it's just going to disappear. Yeah. Okay, so we'll do the white mouse in yellow so that you can see it. So over here, this one's going to be better camouflaged than this one. So this is why the environment is putting pressure on the organisms to become adapted to their environment. So again, if the mice are over here on the light side, the predators are going to see the dark ones more often if the environment was to shift or change in some way, or if they were to move or migrate. Maybe their area, something happens and they've got to move. And so now the population shifts as a result. All right, so what is natural selection? Darwin proposed that natural selection allows populations to adapt to a new environment, and this is the basis of evolution. So natural selection is the process that happens, and it is what is kind of causing this to occur. So Darwin's proposal said that originally giraffe length of their necks varied, that there was some variation in this. And then the struggle to exist causes long neck giraffes to have the most offspring. So the long neck giraffes are able to reach the tree leaves better. They get more food. They're healthier. They're more well nourished. And so they're able to have the most babies. Due to natural selection, most giraffes now have long necks. So what happens is when the long neck giraffe has babies, it passes on the long neck genes, and then all of its offspring that have long necks tend to survive better and produce more long neck giraffes. So hopefully you get the picture about how we have some variation in the population, then there's a struggle to exist, and then due to this pressure on them of trying to get food and compete, we have this change. So then when we talk about variation in populations, we've identified 3 different ways that natural selection can influence the variation for a trait in a population. We have stabilizing selection, directional selection, and disruptive selection. I'm looking at our study guide to make sure that I'm covering the right things. Okay, so stabilizing selection favors the most common form of a variant for the trait and selects against the extreme variants. In this case, the average individual is the most adapted to the environment. So right here, this is stabilizing selection. For directional selection, changes in the environment direct natural selection to favor the variants at one end of the distribution. So just think about the giraffes, how we have a medium neck giraffe, a long neck giraffe, and like a short neck giraffe. So we've got all these variants. So directional selection is favoring the long-necked giraffe. This is causing a shift in the distribution of the trait over time towards one of the extremes, the long neck of the giraffe. So we see that here, directional selection, it's trait over here. Disruptive selection are changes in the environment cause the average or most common form of variation to be detrimental and favors the variance at both ends of the distribution. So here, the average one is problematic in some way, something's happening, and it's favoring the traits at two sides of the distribution. And so now we have disruptive selection. And when we say favoring some variants over others, we're just meaning that these individuals are able to survive and reproduce. Let's see. All right, so our next section here is about how does genetic material change? When we look at DNA, this is our genetic material. This is the genetic material for every organism on Earth, except remember, viruses might have RNA or they might have DNA. But we all have DNA. Our DNA is a double helix. These are considered anti-parallel, so we'll talk about that in a second, and complementary. So the building blocks of DNA is called a nucleotide, and this over here is a nucleotide. So I will circle it for you. This is our nucleotide. In a nucleotide, it is composed of a phosphate, a sugar, and a nitrogen-containing base. So that is what will make it up. These phosphates and sugars will bind together to form the backbone of the DNA. So you can see them over here. So there's that sugar, there's a phosphate, sugar, phosphate, sugar, phosphate. So we often refer to this as the sugar phosphate backbone of DNA. Then in the middle, sticking off of this nucleotide, which I could circle here, let me clear this and then that way you can see it. Here we have our nucleotide, okay? And sticking off the nucleotide, we have the nitrogenous base in the middle. So the nitrogenous bases are complementary to each other. And so what that means is that A, or adenine, will bind to thymine here. So you see A to T. And then guanine on the nucleotide will bind to cytosine. So here's G to C. So that is what we're saying when we're talking about this complementary base pairing with DNA. So guanine to cytosine, adenine to thymine. Then the sequence A's, T's, C's, and G's, these make up the code that is your gene. Okay, so when we have these coding for a protein, we will have a sequence of A's, T's, C's, and G's that code for a specific protein, and that will be the gene. The two strands of the DNA nucleotides are complementary and anti-parallel. So what that means is if I was to separate them, I could get the other strand by looking at the code of one of the strands. So for example, I know that if I have a C, it's going to bind to a G. If I have an A, it's going to bind to a T, and vice versa. If I separated it, I know I have a G here, so I could predict that on the other strand there would be a C. And where I have a T, I could predict on the other strand there would be an A. So that's what they mean by complementary, because they're going to bind with their complementary base pair or complementary base, and that's called the base pairing rule. They are anti-parallel. The DNA has a 5' end and a 3' end, and what it will do is it's shown like this: 3', 5', and we can just pretend this is the DNA. And then the other one that's meeting it will be opposite. So the 5 will be to the 3, and the 3 will be to the 5, okay? So they're anti-parallel. All right, so genetic information is passed from generation to generation through inherited units of chemical information, in most cases, genes. So you get half your DNA from your mom and half your DNA from your dad. You inherited that from them. If there were any changes in the DNA, that could be inherited as well. So when we talk about information flow in biology, normally we go from DNA to RNA to the protein. So we build DNA, we build RNA from DNA in the process of transcription. And then we build RNA into protein in the process of translation. So we've kind of talked about this a little bit. Amino acids are the building blocks of proteins, and each amino acid has a slightly different chemical property. And this, when it's combined, give each protein its specific function in the cell or the organism. They also show you in retroviruses. So remember retroviruses, we said HIV was a retrovirus, and retroviruses are RNA-based viruses, and they take the RNA to synthesize DNA and then do the transcription and translation thing, okay? So if I were to show that, I would be like, we're starting with RNA. Oops, sorry, don't have my pen. So we're starting with RNA, then we go to DNA, then we're going to go from DNA to messenger RNA and then make the protein. I'm doing it again. I'm coloring instead of clicking. All right, now we're going to look at mutations. So mutations are when part of the sequence of the gene changes. So let's pretend that this is a gene. So this is the sequence for the gene. It's C, A, C, G, T, G, etc. Okay, so it's going to be all these little A's, T's, C's, and G's to make up our gene. Each triplet of the DNA codes for a specific amino acid. So they're kind of skipping a step here. Normally we go from DNA to RNA and then to amino acid, but what they're showing you is that the triplet code of this DNA is actually coding for this amino acid, and that's true. So it's going to be read in triplet. And you can kind of go on. So this is valine, histidine or histidine, leucine, threonine, proline, glutamate, possibly glutamate. So that's our sequence of amino acids, okay? So if something were to change here, so here we have this is normally a T, but in this situation, it's being replaced by an A. And that mutates. So there's some kind of error. It looks like we get a completely different amino acid in this situation. So sometimes a mutation is something that changes one of the amino acids. So a change in the nucleotide sequence of a DNA molecule can change the sequence of amino acids in a protein and thus the properties of that protein. So if this valine caused or had significantly different properties than this glutamate, the protein function could be changed and it may not work like it's supposed to, or it may just be doing something completely different. So I'm going to let you guys read through this, give you a minute. So question 8 asks, which of the following processes has the ability to change the genetic information and produce variation in traits? That's going to be mutation. Mutation is going to change the genetic information. It can produce variation in traits. The next question asks, select the statements that are true regarding the process of natural selection and evolutionary change. Individuals vary in their traits, and some of this variation may affect the fitness of the organism. I would say that is true. Over time, natural selection results in populations that are better adapted to their environment. Most of the time, but not always, usually. Natural selection occurs when there is competition for resources. That is true. So we need that competition. And then individuals have the ability to evolve by natural selection over time. That is false because evolution happens at the population level, not individual level. So A, B, and C would be correct. All right, so next we're going to look at lesson 3. I believe this is our last lesson. Um, Now, what causes mutations? And let me go right back to that really, really quick because uh, I didn't discuss that here. So when we look at mutations really quick before we move on, what causes them? It can be as simple as an error in the copying of the DNA. Like I told you, viruses don't have machinery to look at their own errors and fix them. If they do happen to make a faulty or messed up transcription or whatever copy of the DNA. The other thing that can cause mutation is environmental things. So think UV radiation causes mutations in our DNA that can lead to skin cancer. Um, there can be chemicals, things like that, that cause these issues. So could be just as simple as random error, or it could be something environmental. All right, so now over here to what is the evidence that supports evolution? And this is showing you a couple of different species we have. It looks like, I can't tell if this is a butterfly or a moth. It looks like a butterfly. It has wings. And then this owl has wings. And then this other owl has wings. So scientists continue to test, evaluate, and refine their understanding of how evolution impacts life. One of our first evidences for evolution that I've discussed with you are fossils. These are the mineralized remains or traces, trails, droppings, burrows of once living species. Do all organisms form fossils? No. You need special conditions in order to form a fossil. And a lot of times you need hard parts. So some parts of our fossil record are kind of, yeah, because the organisms existing at that time were squishy. And if they were all squishy, they would have had to have left an impression. And the environmental conditions necessary for that were kind of like rare. So when we look at organisms at that time, it's very hard to get a picture of what was happening worldwide or planet-wide, because we only have a couple of areas where this particular type of fossilization occurred. So there very well have been organisms that left no trace, and we have no idea what they look like or what they were. So organisms that lack skeletons and shells may form impressions on the sediments, like I said. Impressions can provide evidence on how life has changed, as well as provide an indication of what the climate and environment were like in the region where they were alive. Some of my favorite impressions are plants. I think like the impressions of bark fossilized from like the Cretaceous are really interesting. So the fossil record gives us a lot of information and shows us how organisms might have looked at different times. One of my favorite things to look at the Houston Museum of Natural Science is the evolution of the horse. The horse started out as a much smaller organism than what we know it to be now. Um, so we see through the fossil record how the horses have changed over, millions of years. Our fossil record also shows this transitional fossils give us information like how species have evolved over time and an indication of the timeline of when these changes occurred. So here we're showing a fish at about 380 million years ago. We also see Tiktaalik, which is kind of this transitional fossil between fish and amphibians. It had some of the characteristics of a fish and some of the characteristics of an amphibian. And then we see the amphibians, which are some of the first tetrapods, meaning four-legged organisms that walked on land at about 360 million years ago. So this is giving us some information, or we're trying to put it together about how did life make it from the oceans to land. And Tiktaalik was a very important fossil because it kind of provided this missing link that we suspected, but needed evidence for. The next one we have in terms of our evidences for evolution, number 2, is comparative anatomy. There are two types of structures. We have analogous structures and homologous structures. So analogous structures serve the same function, they may appear the same, but they evolved independently as adaptations to a specific environment. So the examples are wings of birds, bats, and insects are analogous structures. So they evolved to solve the same problem, which is flight, but they were evolving independently of each other. Okay. So the wings of those organisms all developed in different species at different times to solve the problem of flight, which is a very interesting thing. Homologous structures are structures that are similar because they have been inherited from a common ancestor. The bone structure of the limbs of a mouse, bat, and whale are all similar because these mammals are descended from a common ancestor. So what we'll see is even in you have a humerus, a radius, and an ulna. You've got some carpal bones and some phalanges, finger bones. And the bat has the same layout. It has a humerus, radius, ulna. Its carpals are a little bit different and its phalanges are a little bit different. The dolphin's fin has the same layout, the humerus, radius, ulna, carpals, and then finger bones. So these are homologous structures. These are indicating that they all had a similar or kind of development or common ancestor. The third evidence that we have for evolution is embryonic evidence. Homologous structures are evident during the embryonic development of animals. We can see that embryonic development, we have some similar structures. Not that we are the same, but we have a post-anal tail at a certain point in development, just like the chicks and tortoise. We also have evidence from genetic material, and this one was probably the one that made the biggest impact. So genetic material is not just a set of instructions for various traits, it's a history of our evolutionary past. So we can determine how related we are to other species by looking at the number of differences between our DNA or our genes and theirs. So the closer the genetic information is between the two species, the closer their evolutionary relationship. So humans and primates are the most closely related species because there are very few differences between our genes and theirs. And then you can see as we go on, humans and pigs are a little bit further apart, ducks further, turtles further, fish further, a moth, and then yeast, which is a kind of like microscopic organism, I guess you could say, has a lot of differences. And specifically, they're looking at a gene for cytochrome C. So they can look at genes that we all share, scientists can, and map that gene and then look at the number of differences in the sequence of that gene. And that will tell them how closely related those organisms are. All right, so I'm going to give you a little second to pause here. And again, if you haven't been to the Houston Museum of Natural Science, I highly recommend it, especially for question 10. So question 10, Tiktaalik and Archaeopteryx are associated with what type of evidence for evolution? These would be our fossil evidence, D. Question 11, homologous structures represent which form of evidence supporting the theory of evolution? Comparative anatomy. right, last we have module 2, how do viruses evolve? Lesson 4, how has the SARS-CoV-2 virus evolved? So the fact that viruses can evolve over time presents real challenges for our immune system and for the development of annual vaccines. In 1918, a variant of the H1N1 influenza virus infected over 500 million people across the globe. The death toll worldwide has been estimated to be between 30 and 50 million. This was the 1918 flu, and I believe they estimate that it took out about 1/3 of the whole planet's human population. So this was very deadly flu virus. The SARS-CoV-2 virus has resulted in over 6 million deaths, and that was at the time of the publication of this text. Mostly because of advances in treatment and vaccine development, it's lessened the impact. If we hadn't had that, it would have had a far greater reach. So there are two basic methods by which the SARS-CoV-2 virus changes its genetic information: antigenic shift and antigenic drift. So antigenic drift occurs when there is an accumulation of small genetic changes during replication of the genetic materials. And this is what I was talking with you about how the virus is copying its DNA or RNA in the case of coronavirus, and making copies of itself, and it's making little errors. And so as it's doing that, you put out a virus and it's got one error. Well, then that virus goes and copies itself and makes a virus with two errors. And then that virus copies itself and it makes a virus with three errors. And you keep going over generation after generation. And eventually we have a lot of small genetic changes that are accumulating and causing some very real differences in virus to virus. Okay. Antigenic shift occurs when there is a mixing or reassortment of genes from different viral strains. So antigenic drift is when there are small changes in the surface spikes of the virus. So how is this happening? The virus has to supply its own RNA polymerase to convert viral RNA into messenger RNA. RNA polymerase does not have a high degree of accuracy. It's going to make some errors, so it's going to lead to mutations in the genetic material. Antigenic drift on the spike or S protein of the SARS-CoV-2 is responsible for the formation of variants such as alpha, beta, and gamma. The second mechanism we have is of antigenic shift. This one has greater consequence for humans. So what's happening is in a person, they get infected with two different forms of the virus. So here they've got variant 1 and variant 2, and they're infected. The host cell may manufacture viral particles from both viruses and then reassemble them into a new variant. You might have heard of this. I want to say it was Omicron that they believed had developed in a person who had been co-infected with multiple viruses. So this is something that can occur as well. So let's pause here and you read, and then we'll talk about the answers. So we've got antigenic shift involves what? So let's go back. We've got our antigenic shift is a recombination of viral stuff. So we could say a single host being infected by viruses from two different animals, a formation of new combinations of H and N spikes on the virus, a slower adaptation by the immune system. All of the above are correct. All of these could be the case for that. So the person who was immunocompromised, basically that they said they thought Omicron came, I think it was Omicron. It was one of the variants. I'll have to double check. But if that person's immunocompromised, maybe they're getting infected with several different strains of the virus or different viruses all at the same time, and their immune system isn't handling it. So the viruses are able to replicate in this kind of uncontrolled way. And from there, they can do that whole recombination thing. Which of the following are correct regarding antigenic drift? Select all that apply. So mutations may be introduced by the action of RNA polymerase enzyme. That is correct. Um, it's making errors when it's making the RNA, and that is not fixed. This virus doesn't have a genome big enough to carry around the molecular machinery to uh, spell check itself, basically, is what it is. You have that in your genes, but you have a very big genome. So this virus, it can't pack a lot with it. It really just has to take the essentials. And so it does not take the, uh, I guess you could say, tools it would need to do that. So RNA polymerase makes errors. All right, and that is it for module 2. I hope you enjoyed this long lecture and maybe took some breaks.